Hello students, welcome back to your own channel Vedantu NEET English. This is Baswaraj sir, your biology maths teacher. Quickly let me know in the chat if I am audible and visible to all of you. Quickly let me know each and every one of you. Now students, this is the second part, right? This is the second part for respiration plants, right? So in yesterday's class, we did the first part in which we spoke about what is respiration in general. What is autotrophs, what is heterotrophs. Then we did the beautiful concept of glycolysis, right? Every step by step, every single thing we learned there. Then I also told you something called as respiration sheet after glycolysis. Then we also did fermentation, and that was the end of yesterday's class. Now students, the remaining part, right? The remaining part would be your, what is link reaction, oxidative decarboxylation, what is the meaning of Krebs cycle, and I'll show you a small trick for Krebs cycle, and also I'll teach you what exactly happens in a Krebs cycle, just like how I taught you in glycolysis. No by hearting, understanding it, understanding. Then I'll be teaching you something called as electron transport chain, then amphipoic pathway, respiratory quotient, and the chapter is over. And students, I recommend to watch all of you till the very end. Why? If you watch the session till the very end, you will get the benefit of solving PYQs. That's right. So by the end of today's class, we'll be solving lots and lots of PYQs, which will help you to get the concept clear. For example, whatever you're learning right now in the class, you will know that it is applicable. What is you're learning in the class is useful to all of you. Okay. So with that being said, students, can we start today's class? Can we start? Yes. Let's not waste any time. Let's quickly get into it. And everyone, if you're new to the channel, subscribe to the channel and quickly smash that like button, all of you. And I've been reading your comments in the previous video and it felt really nice to read the comments, let me tell you. Yes, really beautiful comments. Right. Now students, we learned about respiration, anaerobic, aerobic. Now tell me students, I'll ask you a simple question, all of you. Glycolysis, the concept of glycolysis, does it happen in both? Glycolysis, does it happen only in aerobic respiration, or does it happen only in anaerobic respiration, or does it happen in both? Everyone in the chat, answer. Glycolysis, the concept or the process, does it take place only in the case of aerobic? Does it take place only in the case of anaerobic, or both? Everyone in the chat, answer. Or is it both? Quickly, all of you. Or does it happen in both aerobic as well as anaerobic organisms? You will tell me, sir, both. It happens in both aerobic as well as anaerobic organisms. Okay. Now students, under aerobic respiration, tell me, this particular reaction is called complete oxidation. It is called as complete oxidation. Now, this is complete oxidation. Now, where do you notice partial oxidation? Where do you notice partial oxidation? In which reaction or which process? You will tell me, sir, partial oxidation is seen in the case of glycolysis. In the case of glycolysis. What about incomplete oxidation, students? Incomplete oxidation is seen in the case of fermentation reactions. Okay, incomplete oxidation in the case of fermentation. Students, all of these concepts are individual questions. Okay, so don't skip all this. Your incomplete oxidation in the case of fermentation. In the case of fermentation. Okay, beautiful. That is clear to all of you now. Now, here this is aerobic respiration. This is complete oxidation right now. Glucose plus oxygen, energy is being released to carbohydrates plus water. All of you know this simple reaction. Now, all of you are aware that in your glycolysis, pyruvate is made, right? Pyruvate is the end product of glycolysis, is pyruvate. Now, this pyruvate, what does it happen? What is the fate of this pyruvate, right? For example, here we have pyruvate right now. Pyruvate is here. Now, this pyruvate, which is produced in the, where? Cytoplasm or mitochondria? Which is produced in the cytoplasm. All of you would answer. Students, be active in the class. What is this? No energy, already gone. Show some energy in the chat. Whatever questions I'm asking, try answering them. You will all see the benefit of it later on. Okay. For example, I told pyruvate right here. Right, pyruvate. Pyruvate is produced in the cytoplasm. Pyruvate is produced in the cytoplasm. Now, this particular pyruvate will be transported inside the mitochondria. It will be transported inside the mitochondria. Pyruvate is now going inside the mitochondria now. Yes, mitochondria. Now pyruvate is entering. Now once the pyruvate enters the mitochondria, this pyruvate will now undergo 3 different steps. This pyruvate right now will undergo something called as oxidative decarboxylation, link reaction. This pyruvate right here undergoes Krebs cycle. This pyruvate right here later on will undergo electron transport chain. See here. This pyruvate will undergo link reaction, Krebs cycle, and oxidative phosphorylation. Oxidative phosphorylation, which is nothing but electron transport chain. Okay. So this is the concepts which we need to study today. What is link reaction? What is Krebs cycle? And what is electron transport chain, one by one today. Okay. No bioting. We will understand the concept. Let's understand, first of all, let's understand link reaction. Let's understand link reaction. Ready, all of you? Your oxidative decarboxylation is also called as the link reaction here. Link reaction. Link reaction also called as gateway reaction. Gateway reaction. Now, why am I calling it link reaction or gateway reaction? What is it linking? Can anyone tell me what is the linkage happening here? The link which I am talking about here is, this is a reaction which is present between, this link reaction is present between glycolysis and Krebs cycle. So after glycolysis and before Krebs cycle, in the middle there is a reaction that is called as the link reaction or gateway reaction. Now students, understand the beauty of it. Understand the beauty of it. You do not have to by heart this. I will teach you what exactly happens here. First of all, understand the name. It is called as oxidative decarboxylation. It is called as oxidative decarboxylation. So what is the meaning of decarboxylation? The meaning of decarboxylation is release of CO2. Yes, removing of CO2. Yes. So what do we have right over here, students? We have a 3-carbon pyruvate here. 3-carbon pyruvate right here, which is the product of your glycolysis. Now what will happen if I remove a CO2 here? See here, this is a CO2 molecule. Now first step here is remove of CO2. Remove of CO2. That is decarboxylation, means removing the CO2. So I am removing the CO2. Now once you remove the CO2, what am I left with here? Can anyone tell me? What is this COCH3? We are left with COCH3. If you know basic chemistry, can anyone tell me what is the COCH3? The COCH3 is nothing but acetyl group. It is nothing but acetyl group here. Acetyl group right here. Acetyl group. COCH3 is left. Okay. Now students, this COCH3 undergoes oxidation. It undergoes what? It undergoes oxidation. So we have pyruvate initially. Pyruvate, we removed a CO2. We removed a carbon dioxide. We are left with the acetyl group. Acetyl group. Now this acetyl group undergoes oxidation. It undergoes oxidation. Now students, tell me, what is the meaning of oxidation? What is the meaning of oxidation? Can anyone tell me? Oxidation. Oxidation is nothing but, students, I have told you before, oxidation is nothing but loss of electrons or loss of protons. Loss of electrons or loss of protons is called as oxidation. Now tell me, if this acetyl group is getting oxidized, is it losing electrons and protons? It is losing electrons and protons. Okay, it is losing. Now students, this electrons and protons, these electrons and protons are taken up by your NAD+. NAD+. NAD+ will take the electrons and protons. Now tell me students, what do you mean by reduction? What do you mean by reduction? Reduction means gain of electrons or gain of protons, gain of electrons or gain of protons. Now, if this NAD plus right here, if this NAD plus right here is gaining the electrons and it is getting reduced, it is getting what? Reduced to NADH H plus. So what is happening here, students? First of all, the first step is what? The first step is called as decarboxylation. From this pyruvate right here, from this pyruvate right here, a CO2 molecule has been removed. CO2 has been removed. So once CO2 is removed, we are left with the acetyl group. That is the first point. Now this acetyl group undergoes oxidation. What is oxidation? Loss of electrons or loss of protons. Yes. Now once this acetyl group is undergoing oxidation, when this acetyl group is undergoing oxidation, it is this protons and electrons are now taken up by NAD plus. NAD plus. Now this NAD plus, after receiving the protons, it is undergoing what? Reduction. Reduction. So that's how in the link reaction, NADH2 is produced. NADH2 is produced. And at the same time, the third main event here is the addition of coenzyme A. So in the link reaction, what is happening? Pyruvate is undergoing decarboxylation, oxidation and addition of coenzyme A. Now once you add the coenzyme A, we end up getting acetyl coenzyme A. Acetyl coenzyme A, and that is your link reaction. Now this acetyl CoA will now join the Krebs cycle. It is ready to join the Krebs cycle. Student, this right here is the how you understand. This is how you understand the oxidative decarboxylation or link reaction or whatever you call it. Any doubt here, quickly tell me in the chat. Students, any one of you, any doubt, did not understand any single point here? Three main points: decarboxylation, oxidation, as well as addition of coenzyme A. And there is also formation of NADH2. Formation of NADH2 formation is happening. Beautiful. Nice. Now, this is what is I've already mentioned here: decarboxylation, oxidation, and addition of acetyl CoA. Addition of acetyl CoA. Now, students, let's read the NCERT lines to get a better understanding. Let's read the NCERT lines to get the better understanding, all of you. Okay. Now, for aerobic respiration to take place, within the mitochondria, the final product of glycolysis, that is the pyruvate, is transported from the cytoplasm into the mitochondria. So now the pyruvate is sent into the mitochondria. Okay. The crucial events in the aerobic respiration are the complete oxidation of pyruvate by stepwise removal of hydrogen atom and leaving 3 molecules of CO2. This you will learn in the Krebs cycle. Entire Krebs cycle also is mentioned here. Okay. The passing of the electrons removed from as a part of hydrogen molecule to oxygen. This you will learn in electron transport chain. Okay. What is interesting to note that is the first precursor, the first process that takes place in the matrix of mitochondria, while the second process is located in the inner mitochondrial inner membrane of mitochondria. The process which they are telling here, which takes place in the inner membrane, that is nothing but the electron transport chain. That is nothing but the electron transport chain, which takes place in the inner membrane of mitochondria. Clear, all of you in the chat? Now, what about Krebs cycle? Where does the Krebs cycle takes place? Krebs cycle takes place in the matrix of mitochondria. Oxidative decarboxylation link reaction, that also takes place in the case of mitochondria. Clear? Matrix of mitochondria. Only the electron transport chain will take place in the inner membrane of mitochondria. ETC, inner membrane. Now, pyruvate, which is formed by the glycolytic catabolism of carbohydrate, all of you know that, in the cytoplasm, after it enters the mitochondria matrix, undergoes oxidative decarboxylation. See, your pyruvate is undergoing what? pyruvate is undergoing oxidative decarboxylation. All of you know right now what is the meaning of oxidative decarboxylation, right? Now, by a complex set of reactions catalyzed by the pyruvic dehydrogenase is the enzyme. I'll show you that in this thing. Pyruvic dehydrogenase. The reaction catalyzed by the pyruvic dehydrogenase requires the participation of several coenzymes including NAD+. See, NAD+ is receiving the electrons and it is getting converted to NAD+. Okay. Now, this is the reaction. Pyruvic acid, coenzyme A, NAD+. In the presence of pyruvate dehydrogenase, very important, many PYQs have come from here. The enzyme pyruvate dehydrogenase, in the presence of Mg2+, the element, we get acetyl-CoA. A CO2 is released. How? Because of decarboxylation. One NADH is produced as a result of reduction and H+. This is the entire reaction which we learned here. During this process, two molecules. Why two molecules here, students? Why two molecules? Because we know from one glucose we are getting 2 pyruvate. From one glucose we are getting 2 pyruvate. So can I tell from one pyruvic acid we are getting 1 NADH. From 2 pyruvic acid, can I say we'll get into 2? Into 2 this also. Into 2 for NADH. Into 2 for NADH. Yes. Into 2 for NADH. Clear all of you? 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid. That is produced from one glucose. See, from one glucose, 2 pyruvic acid, 2 NADH. The acetyl-CoA then enters the cyclic pathway, tricarboxylic acid cycle. Tricarboxylic acid cycle, also called as Krebs cycle. Tricarboxylic acid or Krebs cycle. After a scientist, named after the scientist Hans Krebs, who first elucidated it. Clear, students? Clear? Now, can we start Krebs cycle now? Yes, can we start the Krebs cycle now, students? Quickly. Tell me in the chat, all of you ready for Krebs cycle? Now, there are 2 methods to learn the Krebs cycle. The first one is very simple. I will tell you a small trick and I will show you how it is done. In 5 minutes, you can learn the Krebs cycle. That is one method. 5 minutes, you can learn the Krebs cycle. That is the first method. The second method which I will be teaching you is, you will be understanding the Krebs cycle, and that will take a lot of time. Because by-hearting is very easy and you can do the by-hearting on your own. But my job as a teacher here is for you to understand and not by-heart it. Like how we did in glycolysis, we understood the glycolysis. Every step by step we made the decoding of it, right? We chopped it out completely. So same thing we'll be doing right now. What we'll do right now is, we will first understand the Krebs cycle in an overline manner. Then we will go to the depths of it. Then we will understand each and every single point. Ready, everyone? Ready? Ready for Krebs cycle, students? All of you know Krebs cycle. The name it is called as Krebs cycle because the scientist who discovered Krebs cycle was called as Hans Krebs. Okay. Understand, students. Understand the Krebs cycle. Okay. Understand the Krebs cycle, all of you. First, I'll tell you a small trick. Officer, can I keep selling sandwiches for money? Understood, all of you? Officer, can I keep selling? Sandwiches for money? The O in the officer, students. The O in the officer stands for oxaloacetate. Oxaloacetate. O in the officer stands for oxaloacetate. Now, what happens here? to this oxaloacetate, students, to this oxaloacetate, which is 4 carbon. This oxaloacetate, which is 4 carbon, we are adding acetyl CoA, which is 2 carbon. Now tell me, students, 4 carbon plus 2 carbon is how many carbons? 4 carbon plus 2 carbon is 6 carbon molecule. 6 carbon. The 6 carbon thing which I am getting here is C. C for citrate. C for citrate. Okay. Citrate. And citrate is 6 carbon. Citrate is 6 carbon. Now, C is done. Now I for isocitrate. I for isocitrate. Isocitrate. Now, next one. K. K for alpha ketoglutarate. Alpha ketoglutarate. K. Now that is K. Ketoglutarate. Now S. What is this S now? The S stands for succinyl coenzyme A. Succinyl coenzyme A. Succinyl coenzyme A. Succinyl coenzyme A. The second S stands for succinyl. The second S right now. See, one is succinyl coenzyme A, other one is going to be succinate. Now, what do we have after that? This is done. F for fumarate. F for fumarate. F for fumarate. Now M for malate. M for malate. And finally, again we are back to the oxaloacetate. Okay, what we'll do is, it doesn't look like a proper circle. 1 second, students. Anyway, it doesn't matter. Now, this is the entire thing. That is, officer, can I keep selling sandwiches for money? That is, O in the officer stands for oxaloacetate, C is citrate, I is isocitrate, K is alpha ketoglutarate, S is for succinyl coenzyme A, second S is for succinate, F for fumarate, M for malate. M in the money stands for malate. Now we have to do some few things here. A few additions and deletions. Few additions basically. Okay. Isocitrate to alpha ketoglutarate, students. In this step right here, there is first, in this step right here, between isocitrate to alpha ketoglutarate, here right now, first oxidative decarboxylation happens. So if oxidative decarboxylation is happening, can I tell there is release of CO2 and there is a release of NADH? Yes. Now between alpha keto and succinyl coenzyme A, the second decarboxylation is happening. Oxidative decarboxylation. Again here one CO2 is released and again there is release of NADH. Now succinyl CoA to your succinate, there is a release of GTP, which is converted later on to ATP. Later on. ATP. Succinate to fumarate, there is a release of water. No, succinate. No. So we have enzyme here. One second. Succinate, right? Succinate dehydrogenase. How can we go wrong here? Now here, before your succinate fumarate, we have FADH2, sorry, not water. FADH2. Fumarate FF. That's how I remember. F for fumarate, F for FADH2. FADH2. Now here water is entering. After here water is entering. Now in this step also, this right here is just oxidation. This right here is oxidation. Okay. This right here is oxidation. If oxidation is happening, we have NADH formation here. NADH formation here. Now here we have decarboxylation also happening. In the here and here in these two places we have decarboxylation. But here right now there is no decarboxylation, only and only oxidation. Now you tell me, students, all of you. Sir, means flower, no? Flower, what flower? I didn't understand. Now tell me. Now here we have 6 carbon. Here also we have 6 carbon. Here also we have 6 carbon. Okay. Here also we have 6 carbon. Now you tell me, if one CO2 is removed here, can I tell alpha ketoglutarate is going to be 5 carbon now? Yes, 5 carbon. Now, if one more CO2 is removed here, succinyl coenzyme A is going to be 4 carbon. Succinate is going to be also 4 carbon. 4 and 4. 2 is entering, 6 again. 6, 5, 4. So that is the thing, right? That is the thing. I can't say it, it's very difficult to say. I can't read very well. Okay. So students, that is the entire flowchart for Krebs cycle. Now, if you can remember this in 5 minutes, right? It doesn't take more than 5 minutes to remember the entire Krebs cycle. Now, students, but it takes a lot of time to understand the Krebs cycle. It takes a lot of time to it's a Pushpa dialogue. First of all, I have to see that movie. First of all, no time to see movies only. So to watch, once I watch the movie, then I'll get to know. See, Pradeep, I can understand Tamil. If you speak, I can understand. Dialogues and all is very difficult to read. Okay. Now, this is the Krebs cycle. Any doubt in Krebs cycle, students? Any doubt in Krebs cycle? Anyone? Any doubt in any part of the Krebs cycle? That's all is Krebs cycle, students. And I'll tell you how the questions will become. They'll ask you questions in which step decarboxylation is happening. In which step GTP formation. There is another name for this. They sometimes in examination they can tell this GTP formation is nothing but this GTP formation is, they can tell it as substrate level phosphorylation. Substrate level phosphorylation, this is right here, is substrate level phosphorylation because ATP is being formed using the substrate. Okay. Clear, all of you? Clear? Such a really good cycle. It's very simple. Now, students, this is what is again is Krebs cycle again. This is what we learned, right? And this is what we learned. First of all, we have citrate, oxaloacetate, 4 carbon, 2 carbon, 6 carbon. 6 carbon, 5 alpha keto. This is 4. From here it is 4. 4 carbon, 4 carbon, and 4 carbon. Right? This is what is happening in your entire Krebs cycle. Officer, can I keep selling sandwiches? Officer, can I keep selling sandwiches for money? M for money. So that is the entire, let's do that's the entire thing. Right? Every single step by step. Beautiful. Now, students, now are you ready for understanding the Krebs cycle now? Tell me. Are you ready to understand the Krebs cycle? See, this is not understanding. We didn't understand the Krebs cycle. This is just the overlay of Krebs cycle. This is just like I've told you citrate, isocitrate. But now I'll be teaching you how it happens one by one in detail. Ready? Now, the first step is what is happening, tell me. In the first step, we have oxaloacetate, 4 carbon. 1, 2, 3, 4. 4 carbon compound oxyl acetate. To this we are adding acetyl-CoA. Acetyl-CoA, that is 2 carbon, right? 1, 2, right? Now in this, say students, in the examination you should know, always learn the first enzyme and the last enzyme. I've always told this. Many questions will come from first and last enzyme always. Now understand here, the first enzyme here is citrate synthase. Please remember that. Citrate synthase you need to remember. Okay. Now when we do that, we end up getting the citrate. Citrate is a 6 carbon molecule. 1, 2, 3, 4, 5, 6. 6 carbon molecule is a citrate. Now understand, this citrate is getting converted to isocitrate. Right. Now this is done. By adding of water. Now citrate to isocitrate. What is happening here is, citrate is isomerized. Now tell me students, what is the meaning of isomerization? Same but different. Right, remember isomerization. Same but different. They have same chemical formula, but structurally they are different. So isomerization is happening. Citrate to isocitrate. Citrate to isocitrate, that is isomerization is happening. Isomerization. Okay. Now we have isocitrate. Now understand, this is a very crucial step now. Very crucial step, very critical step. Understand. All of you remember oxidative decarboxylation, yes or no? Tell me. All of you remember oxidative decarboxylation? Yes, we remember, sir. Now students, understand, this citrate right here, this isocitrate right here, which is after isomerization, it is undergoing oxidation. It is undergoing what? It is undergoing oxidation. Tell me students. Now once it undergoes oxidation, what will happen? Tell me. If a particular structure undergoes oxidation, it will release electrons and release proton. Loss of electrons and loss of protons is happening. Can you see? This structure right here will release electrons and protons. Electrons and protons are released. Now, these electrons and protons are taken up by the NAD+. The NAD+, while taking the electrons, now NAD+ is getting reduced. It is getting reduced to NADH. Getting reduced to NADH. Okay, reduced to NADH. Now, since it is decarboxylation also, there is a loss of CO2. So in this step right here, when I say NADH is produced and CO2 is released out, this is because of oxidative decarboxylation. The first oxidative decarboxylation is happening in the Krebs cycle between isocitrate and alpha-ketoglutarate. First oxidative decarboxylation. All of you understood? Yes. Now that's why this is 5 carbon. Now again, same thing is happening. What is happening now students? From alpha-ketoglutarate to succinyl coenzyme A, once again oxidative decarboxylation is happening. Again, can you see here? Again the same thing is happening. Your alpha-ketoglutarate is undergoing oxidation. It is undergoing oxidation. It is losing the electrons and protons. These electrons and protons are taken up by your NAD+. Where is your NAD+? I think it's there in the next step. Here it is. Next step. It is taken up by your NAD+. And NAD+ undergoes reduction again. So it is going reduction to form NADH. At the same time, there is addition of SH-CoA, succinyl CoA, which is present here. Succinyl CoA has been added. Can you see? So from alpha-ketoglutarate, we are getting succinyl coenzyme A. One of the CO2 is removed completely. There is an addition of CoA. So in this step right here right now, the second oxidative decarboxylation happens. Second time. First time is happening here. Between your isocitrate to alpha-ketoglutarate. Alpha-ketoglutarate to succinyl coenzyme A, second one is happening. Okay. Now understand students. One minute again. Which one did you understand? Which one did you understand? See here, understand this very carefully. All of you know what is oxidative decarboxylation, right? First of all, we learned it here. We learned it here in the link reaction, all of you. Let's go back to ring reaction and I'll show you. We learned it here also. We learned it here. We learned it here. Oxidative decarboxylation. The meaning of oxidative decarboxylation is that there is a release of CO2. Decarboxylation. What is oxidation? The structure is getting oxidized. When a structure gets oxidized, it will lose electrons and protons. It will lose electrons and protons. Oxidation, loss of electrons and loss of protons. That particular electrons and protons are taken up by the group. Here, electrons and protons are taken up by your NAD+. If NAD+ is receiving electrons, if NAD+ is receiving the electrons and protons, can I tell it is undergoing reduction? Gain of electrons and gain of protons. Going reduction to produce NADH. The same thing is also happening. The same thing is also happening in the two steps of glycolysis. In the two steps of glycolysis. Between isocitrate to alpha ketoglutarate. Between isocitrate to alpha ketoglutarate. Can you see here? Between isocitrate to alpha ketoglutarate. What is happening here? Your isocitrate is undergoing oxidation. Oxidation. Tell me, what is oxidation, students? The meaning of oxidation is loss of electrons or loss of protons. True. Now, this particular structure, it is undergoing oxidation. It will lose electrons and it will lose protons. Yes. This electrons and protons lost by the isocitrate is picked up by NAD+. Picked up by NAD+. Now, if NAD+ is taking protons, it is taking electrons, can I tell it is undergoing reduction? Yes or no? What is reduction, students? What is reduction? Reduction is nothing but gain of electrons and protons. So can I tell this particular NAD+ is undergoing reduction to produce NADH. Now, since this step is called as what? This step is called as oxidative decarboxylation. There has to be removal of CO2. Decarboxylation. Carboxylation is basically the CO2. D means removal of CO2. So can I tell? One CO2 is removed. This was 6 carbon. So 6 carbon will become what? 6 carbon will become 5 carbon. Why? Because 1 carbon is lost as CO2. It is lost as a CO2. This is called as oxidative decarboxylation. And the same step is happening twice. Once it is happening between isocitrate to alpha ketoglutarate. The same oxidative decarboxylation is happening again second time between alpha ketoglutarate and succinyl coenzyme A. Same thing is happening. Can you see? Here to here. NADH is produced, CO2 is removed out. With the addition of CoA. Now, students, this succinyl CoA is like all of you, very unstable. Morning you will open physics, within 10 minutes you will close the physics and you open biology because you like biology. Suddenly you will close biology, you will open chemistry. Very unstable, don't know what to study. Okay, so just like all of you, this succinyl coenzyme A is very unstable. Succinyl coenzyme A is very unstable here. Okay. This succinyl coenzyme A, what will happen is, it will form a intermediate product. It will form a intermediate product called as succinyl phosphate. Intermediate product. When it is forming the intermediate product, there is addition of phosphate here. And this coenzyme A, can you see this coenzyme A part is removed. It is removed. So we get a structure like this, succinyl phosphate. Now again, this also is very unstable. Succinyl phosphate. So what will happen is, it will lose, since it is very unstable, it will lose the phosphate. This is removed. And this particular phosphate which is removed, it's taken up by GDP to make GTP. Guanosine diphosphate will become If you add this phosphate into GDP, it will become GTP. And finally, from succinyl coenzyme A, we are getting a stable succinate, a very stable succinate. I believe all of you understood the concept here, right? How you get from succinyl coenzyme A to succinate because of addition of a phosphate, removal of CoAsh, and we get an intermediate product called as succinate phosphate, which is not given in cycle. This intermediate is not given in cycle. Okay, see here. Directly we have from succinyl CoA to succinate with the release of GTP. Now all of you know how GTP is made. All of you know how this GTP is coming into existence. All of you know how GTP is coming into existence. Eventually, students, this GTP will be converted to ATP. Converted to ATP. Clear? Clear? Now, succinate of fumarate. Succinate of fumarate is normal. Same thing, oxidizing happening. It is undergoing oxidation. It is losing electrons and protons. Electrons and protons are taken up by FAD. FAD is reduced to FADH. But it is simple oxidation. It is just oxidation. It is just oxidation. It is not oxidative decarboxylation. Decarboxylation only happened twice. Right now it is a simple oxidation. Simple oxidation, FAD is getting reduced to FADH. That's why between succinate of fumarate, as a result of oxidation, we have the FADH2. We have the FADH2, all of you. Now, fumarate to malate, adding of water. Fumarate to malate, water is being added. Can you see a water molecule? H2O has been added here. H2O has been added here. Yes, here and here. H2O is added here. Water has been added. Now, same thing happens. This malate will undergo oxidation. Oxidation will undergo oxidation, and it will release electrons and protons. Electrons and protons are taken up by your NAD, and it will becoming NADH. But again, is this decarboxylation? No, it is not decarboxylation. It is a simple, very simple oxidation. Very simple oxidation, not decarboxylation. The decarboxylation will happen only how many times? Only 2 times. Once here and second here. Decarboxylation will happen. Clear, all of you? Did you understand Krebs cycle? See, if you see from outside, it looks very simple. It looks very simple and easy. You know the trick also to remember it. But when you study in detail, you will understand the concept that this entire thing, this entire process is basically powered by oxidation reduction. Yes or no? Tell me, students. This entire process is basically energy is given by oxidation reduction. There is oxidation happening, reduction happening. Oxidation, reduction. Oxidation, reduction. Entire redox reaction keeps on happening here. Redox reaction keeps on taking place here. That is the concept which I want you to take away today's class. That is the concept which you need to take away. Redox reactions are happening here. Okay. This is your entire cycle, student. This is your entire cycle of Krebs. Beautiful, right? Beautiful Krebs cycle. Nice. Ready for some question? All of you. Ready for one question? I'll ask you. Look at this cycle. And can I ask you a question from this cycle now, all of you? Can I ask you some questions based on this cycle right here? Based on this cycle, can I ask you some questions? Okay, I'll ask you. I'll give you 10 seconds or I'll give you 20 seconds. Read the entire cycle and I'll ask you what exactly happened where. Ready? Ready, all of you? Ready? 1 second, students. Ready? Okay. Now. Tell me, in which step, identify the step in tricarboxylic acid cycle which does not involve oxidation of a substrate. Which does not involve oxidation of substrate. Read the question and tell me the answer. There is no oxidation. succinic acid to malic acid, succinic to malic acid, succinyl coenzyme into succinic acid, isocitrate to alpha glutarate, malic acid to oxaloacetic acid. What is the answer? I'll give you a clue. I'll give you a clue, students. The clue here is in the step, right? In that step, GTP is produced. GTP is produced. Remember, GTP is produced as a result of substrate level phosphorylation. GTP is produced as a result of substrate level phosphorylation. Rest everything is produced as a result of oxidation. Tell me the answer. Tell me the answer now, students. In which step GTP is produced? If you go back here, between your succinyl-CoA to succinate, in that step GTP is produced. It means here, what is happening here? In this step right here, students, in this step right here, substrate level phosphorylation is happening. There is no oxidation. There is no oxidation. So do you realize this was a NEET 2024 question? And more than 50% students went wrong here because they could not understand the question only. Why? Why were you not able to understand the question? Because you just by-hearted the Krebs cycle from outside and didn't understand the Krebs cycle. Clear, all of you? So only when you understand the Krebs cycle, you can appreciate all this. You can solve such questions. You can solve such questions. Okay? You can solve such questions. Otherwise, it's very difficult to solve such questions. Okay. Now, students, see here. In your entire Krebs cycle, they have given you one table here. Okay, one small reaction. Pyruvic acid plus 4 NADH, 1 FADH, 2 water, ADP in mitochondrial matrix. We are getting 3 CO2, 4 NADH, 4 H plus, 1 FADH and 1 ATP. Now my question is, in the Krebs cycle, we learned right now, here we have 1 NADH, here we have 1 NADH, 1 CO2, 1 CO2 and 1 more NADH here. So in total there are only 3 NADH and 2 CO2 and 1 GTP and 1 FADH. Why do you think in your NCERT it mentions as 4 NADH and 3 CO2 instead of 2 CO2? Why it is mentioned 3 CO2 and why it is mentioned as 4 NADH? Can anyone tell me? Anyone? Why do you think they have mentioned 1 FADH? All of you know 1 FADH, all of you know. Why 4 NAD plus? Why? Because this reaction right here, student, this reaction right here is happening. They have given for entire mitochondrial matrix. This reaction is right now mitochondrial matrix, and all of you should be aware that in the mitochondrial matrix, oxidative decarboxylation is also happening. So this reaction right here, this reaction is right here is nothing but link reaction plus Krebs cycle plus the Krebs cycle. In the link reaction we learned, right? In link reaction we learned that there is one NADH is being produced in link reaction. In link reaction also there is one CO2 being produced. That is why all of you should know link reaction is also called as oxidative decarboxylation. And that is why they are given pyruvic acid here. If this was Krebs cycle, they should have given acetyl-CoA. But they have mentioned pyruvic acid here, not acetyl-CoA. So they are trying to mention here, this paragraph, this particular line here, mixture of both. Okay, link reaction and Krebs cycle. Now students, can we read the NCERT now? Can we read the NCERT now? All of you? Can we start? Can we start, students? Yes. The TCA cycle starts with the condensation of acetyl-CoA group with oxaloacetic acid and water to yield citric acid. We learned that here. Acetyl-CoA, oxaloacetic acid, water and citrate. We end up getting citrate. Okay, citric acid. The reaction is catalyzed by an enzyme, citrate synthase. See, I told you. First enzyme they always mention Citrate synthase, very important. Very important. PYQ. PYQ. This also is a PYQ. Citrate synthase. And a molecule of CoAsh is released. It is released out. Citrate is then isomerized. See, isomerization. Second step is called as what? Isomerization to isocitrate. It is followed by two successive steps of decarboxylation. Two steps of decarboxylation. Okay, two steps of decarboxylation. Okay, decarboxylation, leading to the formation of alpha ketoglutarate, which is 5 carbon, and succinyl coenzyme A, which is 4 carbon. Now, in the remaining steps of citric acid cycle, succinyl coenzyme A is oxidized to OAA, allowing the cycle to continue. Now, during the conversion of succinyl coenzyme to succinic acid, a molecule of GDP is synthesized. This is a substrate level phosphorylation. All of you learned substrate level phosphorylation. Yes. In a coupled with GDP is converted to GDP, while simultaneously synthesis of ATP from ADP is also done. Okay. Also, there are 3 points in cycle where NAD plus, all of you know, is reduced to NADH. All of you know the reduction process now. What is reduction here? Reduction is nothing but gain of electrons or gain of protons. Right. Reduced to NADH. And at one point where FADH is reduced to NAD, FADH2. That is happening between fumarate to malate. Fumarate to malate. Yes. Now, the continued oxidation of acetyl-CoA via TCA cycle requires continuous replenishment of the OAA. Right? Obviously, if the cycle has to continue, it requires more and more OAA. And the first member of the cycle. In addition, it also requires regeneration of NAD plus and FAD plus, because it is getting consumed everywhere from NADH and FADH2 respectively. The summary equation for this phase of respiratory phase be written as follows. It is given here. This is the summary. Okay. Now, we have till now seen the glucose is now being broken down to release CO2 and 8 molecules of NADH. 8 molecules of NADH till now. Okay. 8 molecules of NADH from one glucose. In the glycolysis 1, glycolysis 2 NADH. Now here we have 3 NADH, right? Totally 3. 3 into 2 is 6. See here, in glycolysis, we had 2 NADH. And in the Krebs cycle, in the entire cycle only there were 3. In the entire cycle only there were 3. 3 into 2 is 6. 6 NADH. But here right now what they are mentioning here, 8 molecules of NADH, it does not include the glycolysis one, it actually includes the link reaction one. Link reaction also, how many? 2 NADH. Don't worry, in the end I will tell you this thing. In the end I'll tell you what is that entire net gain and everything. You will understand the entire respiratory balance sheet there. Okay. Now, and 2 molecules of FADH have been synthesized beside the 2 molecules of ATP in the TCA cycle. Okay. 2 molecules of ATP, because it is happening twice for one glucose. Okay. You may be wondering why we have been discussing respiration at all. Neither O2 has come into picture nor promised large amount of AT has been synthesized yet. True, right? Till now, students, in the glycolysis, in the link reaction, in Krebs cycle, did you see anywhere large amount of ATP being produced? No. There was NADPH formation, 4 ATP in glycolysis, 2 ATP in Krebs cycle. There has been no place till now where large amount of ATP is produced. Yes, large amount of ATP is produced. Never. Till now, have we ever encountered glucose anywhere? Have we encountered anywhere oxygen anywhere till now? No. Why? Tell me, in the end of the movie only, the hero entry will happen once again, second time. Second time the hero entry will happen right in the movie towards the very end with powerfulness. The hero is still coming. The real hero of this movie is still coming. That is oxygen. And the role of oxygen in the entire respiration, the role of oxygen in the entire respiration will happen only towards the very end. Only and only towards the very end. Until that end is here, oxygen is not entering anywhere in aerobic respiration. Wait for it. Okay. Now, also, what is the role of NAD? What is the role of NAD and FADH2 that we synthesize? That is, what is the fate of this NADH? What is the use of this NADH and what is the use of this FADH that we synthesize? Let us try to understand the role of O2 in respiration and how ATP is synthesized. That is coming towards the climax now. Ready, students? We are coming towards the climax of respiration now. Climax of respiration. Last step in respiration. Technically, the last step in respiration. Ready? All of you in the chat. That is, what is the role of NADH and FADH2 that is synthesized here? Like we got NADH, we got FADH in the Krebs cycle. What is the actual role of all this? Where is it being used? Where is it being used? And where is this? Where is the stupid oxygen till now? Till now, the oxygen entry has not happened yet, right? So the oxygen is coming now. Wait. For that, you need to learn something called as the last step of the puzzle. The last step of this reaction is called as oxidative phosphorylation. What is the meaning of phosphorylation, students? Meaning of phosphorylation usually refers to the formation of ATP. Meaning of oxidative here is using the oxygen. Using the oxygen is involved here. Okay, climax. The NADH2 and FADH2 are transferred to the electron transport chain. The ATP is formed as a result of transfer of electrons. Okay. Now here, this is a matrix. This is a matrix. That is the matrix. This is the outer membrane. This is the outer membrane here. Outer membrane. Now, students, in this inner membrane, in the cristae, do you see this cristae? In this inner membrane, the final reaction is happening. The final reaction is happening in that particular cycle there. See here. That. So Krebs cycle is happening where? Krebs cycle is happening in the matrix. But on the membrane, inner mitochondrial membrane, we have the electron transport chain happening. We have the electron transport chain happening. Students, let me know in the chat, can we start electron transport chain now? The last part of today's class. Let's see, after this we have something else. After this electron transport chain, we have amphiboic pathway, respiratory quotient. But all that is very small topic. All that is very small topic. The main topic for today is electron transport chain. Let me know, can we start that right now? All of you drink some water and come. All of you drink some water and come. And we will start electron transport chain. So by that time, I will also go. I didn't get my water right now. So I'll go get my water. By the time I go get my water, I want everyone to draw this. I want everyone to draw this. So draw this and keep. I will go and get my water. Okay? Quickly draw this. You will understand electron transport chain now, students. You will understand the electron transport chain. Quickly draw all of this. Yes? Draw all this. Draw this and keep. All of you. Now students, can we start now? Tell me. Can we start? The electron transport chain, the last part of today's class, and trust me, you will enjoy it. This is very interesting to learn everything. And now I'll teach you in a very easy manner. You will learn electron transport chain, right? Learn electron transport chain, and if you practice it enough, you will never forget it. Okay. Now, before we learn electron transport chain, I want everyone to write down. Something called as protein complexes. I want to write down something called as protein complexes. Protein complexes are nothing but they're made up of protein. They're made up of protein plus certain electron carrier. Electron carriers. Here we have something called as your complex 1, complex 2, complex 3, complex 4 and complex 5. 5 different complexes. For example, complex 1 is called as NADH dehydrogenase. Complex 2 is called as succinate dehydrogenase. Complex 3 is called as cytochrome BC1 complex. Complex 4 is called as cytochrome oxidase, and complex 5 is called as ATP synthase. 5 different complexes which you need to know if you want to understand the entire cycle. And students, is this important to write everything? Yes, it is important. Why? Because in examination there has been a question where they tell you which is complex 1, which is complex 2, which is complex 3 and which is complex 4. They have asked this question previously, so it is very important to learn that. Okay. Now students, now that is your complex. I'll repeat again: NADH dehydrogenase, succinyl dehydrogenase, cytochrome BC1 complex, cytochrome C oxidase, as well as ATP synthase. Now, what is this electron mobile carrier now? Mobile carrier are like your taxi, auto, right? Whenever you want to go from your house to your friend's house or your house to your school or somewhere, you take the auto and you go to the destination. Take the auto and travel. Now here also, these mobile carriers are basically autos for the electrons. If the electron has to travel somewhere, it will not walk on its foot. It will open the app, order an auto, sit in the auto and then go. That is called as mobile carrier. So we have something called as ubiquinone and cytochrome C oxidase. Both of them are electron carrier. Especially this ubiquinone, students. When this ubiquinone takes up the electrons and protons, it gets converted to ubiquinol. That is, UQ will get converted to UQH2. That is ubiquinol. Okay, ubiquinol. Now understand, students, understand. Ubiquinol. Okay. Now I will try to fill the entire complexes. Follow me one by one and fill the complex with me. Now students, the above part is called as a matrix. Now tell me, in the matrix what happens? In the matrix we have the Krebs cycle taking place. Yes. In the matrix, can I tell? In the matrix right here we have the Krebs cycle. So can I tell in this Krebs cycle we have formation of NADH and also FADH2. Formation is happening in Krebs cycle. Yes, it is happening in the Krebs cycle. This right here is the intermembrane. This is inner membrane of mitochondria. Then we have outer membrane. This is intermembrane. Between that. Okay. Now let's fill all the following one by one. This here is called as complex 1. This is called as complex 1. This is called as complex 3. This is complex 2. This is complex 4 and this is called as complex 5. Okay, complex 5 is there. Now understand what is happening. Let's fill each one of them. This complex 1 is called as NADH dehydrogenase. So we have FMN and we have your iron sulfur centers. Your complex 3, BC1 complex, right? BC1 complex. So we have cytochrome B C1 complex, and also iron sulfur centers. The second one is called as FADH dehydrogenase or succinate dehydrogenase, right? Succinate dehydrogenase. So it's also called as FADH dehydrogenase. FAD dehydrogenase. So FAD and iron sulfur centers. Now the last complex, the complex number 4, last but one, is called as your cytochrome oxidase. So here we have something called as copper center A, copper center B, cytochrome A1 and cytochrome A3. Clear? The last one here, this is the F0, this is the F1. F0 and F1. Okay? F0 and F1. Now understand what is happening here. Understand what is happening, students. Now, this is the first complex right here. Now, in this first complex, we have the NADH is landing here. Your NADH is landing here. NADH2 is landing here. Now, this NADH2 is undergoing oxidation. What is oxidation, students? Oxidation is nothing but loss of proton or loss of electron. So, it is getting oxidized to NAD+. Similarly, here also. Here in the complex 2 we have the FADH2, which is oxidized to FAD+. Okay. Now, students, what is happening is your NAD+, which is getting oxidized, it is losing electrons. Electrons are sent here, and electrons are being sent here. Yes, electrons are being sent. It is losing protons. Yes, it is losing protons. So can I tell protons are sent here? 4H+. Technically, just 2H+ first. 2H+. Protons are sent here. Where? From here. Directly protons are also sent. Protons are also being sent here. Okay. H+ is being sent here. At the same time, students, this particular complex can also do its own work, and it can take 2 protons from the matrix and send 2H+ again down. So in total there are 4H+ from complex 1. 2H+ because of your NADH, and one more 2H+ because the complex itself is sending 2H+ from the matrix into the intermembrane space. So 4H+ is coming here. Okay. Now, students, here we have a mobile carrier. Now we have the mobile carrier. We have the mobile carrier called as UQ here. UQ. Now UQH2. Okay. Now, students, these electrons are now given to UQ. UQ will cycle the electrons, receive the electrons, and become UQH2. And this cycle keeps on continuing. Now, how is it becoming UQH2? Because it is able to pull 2H+ from the matrix. Able to pull 2H+ from the matrix. 2H+ from the matrix. Okay. Able to pull. Now, what is happening here? 2 electrons are added. 2 electrons are being sent here. 2 electrons added from here. Now, from this UQ to your complex 3, 1 electrons are being sent. 1 electron is being sent. 1 electron is being sent here. Okay. To your complex number 3. Complex number 3, cytochrome CBC1 complex. Okay. Now understand. Now you need to understand one small concept here, students. Can you see your complex 1, complex 3, complex 4 and complex 5? All the following complexes are intermembrane complex. Intermembrane. They are coming all the way to the intermembrane. But if you notice, students, your complex 2, it is stopping in the inner membrane only. This complex 2 is not coming to the intermembrane. So your complex 2 at FADH dehydrogenase, right, or succinate dehydrogenase, it is stopping at inner membrane. It is not coming down. So technically what is happening is, when it is losing electrons and protons, the protons are not able to come here. The protons are not able to come through this. Why? Because there is no space, there is no gap, as in like there is no way for it to come out here. There is no way for it to come out here. Okay. So what happens is, here also we have UQ and ubiquinol, ubiquinone and ubiquinol, NOL, NOL. Okay. So what is happening here is same thing. This FAD plus is getting oxidized to FAD. What is oxidation? Tell me, students. Oxidation is nothing but loss of electrons or loss of protons. Oxidation is happening. Oxidation is happening. So it is losing electrons and protons. Electrons and protons. And these two electrons are given to UQ. UQ. Now UQ will run. It will run as same cycle. Same cycle will run right now. 2 electrons. Yes. 2 electrons are entering. Second. Okay. 2 electrons again given here. UQ. Same cycle will run now. And from here also, electrons are being sent here. That is how many electron? 1 electron. 1 electron. Okay. Understand now. Understand, students. Now, this 2H plus, which is received by UQ, the 2H plus is also sent here. Is also sent here. From here also we are getting electrons and 2H plus, right? 2H plus. From here also, if it is getting oxidized, we are getting electrons and 2H plus. 2H plus, right? Electrons are sent here, and this 2H plus will be again taken up by this UQ. UQ. And this UQ will also send the 2H plus. 2H plus to complex 3. Complex 3. Now you might ask me, students, you might ask me, sir, here there is only 2 electrons are entering. 2 electrons are entering. But only 1 electron is given here. Why? Because, students, 1 electron is being used to run the cycle. To run the cycle here. To run the cycle. Okay. Now what do we have, students, here? What do we have? Right now we have in the complex 3, 2 electrons, and 2 electrons. What about the protons? Right here, if you notice, 2H plus, 2H plus. So can I tell 4H plus is out here also? 4H plus is also out here. Okay. 4H plus is also out here. Beautiful. Now this particular 2 electrons, has to go to the complex 4 now. Tell me, how can it go? Can it take a taxi or auto? Yes, it can take auto now. The auto here is in the form of cytochrome C is the auto. The cytochrome C will pick up the electrons. It will pick up the electrons, and this is transported to complex 4. Cytochrome C. Now the 2 electrons are entering here, entering 2 electrons, entering, and finally coming out. 2 electrons. Now, students, who was the main hero of the story? Tell me in the chat. Who was the main hero of the story? All this while, in the entire, when you write the equation of your glycolysis, when you write the equation of your write the equation of respiration, do we not always write C6H12O6 plus oxygen will give you CO2 plus water? Do we not write this always, students? Do we not write this always? Plus energy. But till now the chapter is coming to the end. Till now the chapter is already coming to the end, and there is no way oxygen is seen. And nowhere I saw formation of water till now. Nowhere. Till now it is not there. Okay. Now is the hero entry, students. Now is the hero entry. The final electron acceptor, students. I repeat again. The final electron acceptor. The final electron acceptor is none other than your oxygen. Final electron acceptor, students. Finally, it is here. Finally, it is here, students. Finally, it is here. Finally, it is here. Properly. Will give you water. Will give you water. And that is how, students. That is how in the entire respiration, the final electron acceptor, the role of oxygen in the respiration, is only towards the very end. Only towards the very end is the role of oxygen. And that is how towards the very end there is the release of water also. Release of water. So when this is happening, when this entire process is happening, from the matrix, using the electron energy, 2H plus is sent into the intermembrane space. Now tell me, can I tell right now in the intermembrane there are in total of 10 H plus right now? 10 H plus right now? Yes, 10 H plus right now. And this 10 H plus right now here, which is present in the intermembrane space, the 10 H plus present here, this 10 H plus, out of this 10 H plus, every 4 H plus which goes from here, will end up will convert 1 ADP, into 1 ATP. 1 ATP. Every 4H plus will make 1 ATP later on. 1 ATP later on. Clear, all of you? Tell me, students. Clear? Clear? Four H plus will make 1 ATP. Now, students, I will quickly summarize everything. I'll quickly summarize the entire thing for you. I know if you read this entire, it may look at this. It looks like a work of art, right? It looks like a work of art. Just look at it. Just appreciate this, students. Just appreciate the entire cycle, how it is happening inside your body. This is also happening inside your body, students. I will revise it one more time for all of you. Just listen to what I say. Okay, all of you. Take a screenshot or whatever you want. But if you want the notes, all of you know, right? I will upload these notes and other remaining notes on the Telegram channel. I'll be uploading the notes here. Okay. Now, I will just revisit every single topic and you just follow through. Okay. Now, students. No. First of all, this is matrix, inner membrane and intermembrane. Now, complex 1, complex 3, then complex 2 and complex 4 and complex 5. Okay. Complex 1, NADH dehydrogenase. Here we have the NADH is being converted to NAD+. What is this? Simple oxidation. And whenever there is oxidation, there is the release of electrons and release of protons, and same electrons are taken in and protons are removed out here. And same one more 2H plus is removed out because of the entire mechanism here. The NADH dehydrogenase, the enzyme itself removes 2H plus. Okay. Now, totally 4H plus here. Now electrons are now given to UQ. UQ is a mobile carrier. UQ will take the electrons, take the protons, get reduced to UQH2. Again the cycle will keep on continuing. And that's why only one electron is sent here. One electron is used to continue the cycle. And at the same time, 2H plus from the matrix is used by UQ2, and the same 2H plus is given to the complex 3. It is given to the complex 3. Similarly, at the same time in complex 2, what is happening is, we have FADH2, it's getting oxidized to FAD plus. When this oxidation is happening, 2 proton and electron are taken. 2 proton and electron are given. This proton and electrons are sent to UQ again. UQ will use 1 electron to run the cycle, and 1 electron and 2 protons are sent to your complex 3. Together, the 4 protons are out here, and at the same time, the electron also is out. But now, the electron right here, the electron has to travel to the complex 4. How will it travel? With the help of mobile carrier, cytochrome C. Now, once it reaches here, the electrons are moved up. And finally, we have the final electron acceptor, that is oxygen, the role of oxygen to receive electrons, that is the glucose is here. So oxygen is here. Okay, oxygen. Oxygen, after receiving the electrons, is getting converted to water molecule. So that is why they say water is a byproduct of your respiration. Then students, once we have enough of protons here, every 4 protons we get 1 ATP. That is 3 protons, students. That is students, if you notice here, that is 1 ATP we need at least 3 protons. First of all, we need 3 protons to rotate the ring. We need 3 protons to rotate the ring. One more extra proton is used to bring this inorganic phosphate and attach it here. And only when 4 protons are there, 4 H plus are there, 1 ATP is made, not 2. In NCERT, they still write 2 here. I don't know why. They write 2 here. This is supposed to be 4H plus actually. Supposed to be 4H plus. Okay. 4H plus will make 1 ATP. Clear, all of you? Clear? The same thing is given here, students. Same thing is given here. Complex 1, complex 3, complex 2, complex 4, complex 5. Same centers, FMN, FES. BC1 complex, FES. FAD and copper centers. Copper center A, copper center B, cytochrome A1, cytochrome A3. Here we have fumarate to malate, it is FAD is produced here. NADH is given here. 4H plus and 2H plus. Electron carrier, can you see from here? Now here in the middle, can you see? UQ, UQH2, UQ, UQH2, right? Exactly. Serotonin, it is the opposite in the case of chemiosmotic hypothesis. In the lumen, more number of protons are there. In the chemiosmotic hypothesis. Okay. The same thing, what is given here in the vertical manner, I have drawn it in a horizontal manner here. For you to understand better. Okay? Friends, trust me, this is the really concept wise, this is a real concept. And if you want to understand this entire thing, right, it will take time. Right? You might have understood right now, but unless you write it yourself and practice it at least 2 to 3 times, then only you will be able to master this. Unless you do that, very difficult until then. So try mastering this while writing it more and more often. It may look complicated right now. Can you see? It looks very complicated, right? Matrix, intermembrane, so many different colors, so many different things are happening. But it is actually very easy if you practice it enough. Okay. Now students. Now, respiratory balance sheet. Ready? Ready now students? For respiratory balance sheet, whatever we have learned in this chapter, we'll put it in this one sheet right now. In one slide, we'll put everything what we have learned here in this entire chapter. Okay. Let's do the respiratory balance sheet. Okay. Now, let's divide it into 3 categories. The first one we have glycolysis here. First one we have the glycolysis. Glycolysis is there. Then we have the link reaction. Link reaction. Then we have the Krebs cycle. Then we have the Krebs cycle. Then we have the grand total. Then we have the grand total. Then we have the grand total. Okay. Now students, tell me, in the case of glycolysis, how much ATP do you get? In the case of glycolysis, first we get -2 ATP. -2 ATP. Then we get something called as 2 NADH, which is 6 ATP. 6 ATP. And then we also get in the last 7th step and 10th step, we get 4 ATP. So in total, there was gross was how much? Gross was how much here? Gross was 10 ATP. Net was how much? 10 minus 2 here. So 8 ATP we got from 8 ATP we ended up getting from glycolysis. 8 ATP from the glycolysis. 8 ATP from glycolysis. Clear? Any doubt in this one? Tell me. Any doubt in this one? 8 ATPs. Right? 8 ATPs from the glycolysis. Now the second one called as link reaction. Students, in the link reaction, was there any ATP formation? Tell me. In the link reaction, was there ATP formation? No. In link reaction, there were 2 NADH produced. 2 NADH. So can I tell 1 NADH gives how many ATP? 1 NADH gives you 3 ATP. And 1 FADH gives you 2 ATP. So can I tell 3 2 is 6 ATPs here? Grand total. 6 ATPs here? Yes, 6 ATPs here. 8, 6. Now, Krebs cycle. Let's do Krebs cycle. In Krebs cycle, students, how many NADH do you get, students, totally? Tell me, how many NADH first of all you get? NADH. All of you know there are 3 NADH in one cycle. 3 NADH in one cycle. But I am writing for 1 glucose here. For 1 glucose. For 1 glucose I am writing here. So for 1 glucose, the cycle has to go twice. The cycle has to go twice. So in the Krebs cycle, which we know, the 3 NADH you get. The 3 NADH you get is as the result of 1 cycle. 3 NADH is the result of 1 cycle. That is from 1 pyruvate or 1 acetyl-CoA. 1 acetyl-CoA. So if the cycle is happening twice for 1 glycolysis, so can I write 6 NADH? 6 NADH. 6 NADH. How many ATP, students? In 1 cycle, 1 GTP. 2 cycles, 2 GTP. In 1 cycle, 1 FADH. 2 cycles, 2 FADH. Six into 3, this is into 1 as usual, this is into 2. That is nothing but 18 ATP here, 18 ATP, 2 ATP from here, and 4 ATP from here. 4 ATP from here. Now, what is the total? Tell me, students, what is the total now? Total. 8 plus 2. 8 plus 2 is 20. 20 plus 6, 26. 26 plus 4 is 30. 30 plus 8 is 38. Done. Tell me, students, 38 ATP is the total. 38 ATP is the grand total. 38 ATPs is the grand total which is happening in the entire glycolysis. Entire glycolysis. 38 ATPs. 38 ATPs from one glucose. From one glucose. 38 ATPs. Clear, all of you? Clear? Clear, students? 38 ATP from one glucose. Let's see what is given in NCERT now. NCERT says it is possible to calculate the net gain of ATP for every glucose molecule oxidized, but in reality this can only remain a theoretical exercise. So what we did right now, it is theoretical exercise. It is theoretical exercise. Okay. Okay. Now these calculations can only be made on certain assumptions. There is a sequence orderly pathway functioning with one substrate forming the next within the glycolysis. TCA cycle and ETS pathway following one after another. See students, how did we learn the chapter? We learned the chapter in an orderly fashion. First we did glycolysis, then Krebs cycle, link reaction, and then electron transport system. But in reality, they will not happen one after another. They will be happening simultaneously. Right? The NADH synthesis in glycolysis is transferred into mitochondria and undergoes oxide decarboxylation phosphorylation. All of you learned that. See, this is used in glycolysis. I am telling you, this NADH is used in the electron transport system. Okay. None of the intermediates in the pathway are utilized to synthesize any other compound. See, intermediates, we got so many intermediates right now. We got so many intermediates, but we never use any type of intermediate right now. We did not use any intermediate. Right. Only glucose is being respired. No other alternative substrates are entering into the pathway at any of the intermediate stage. Because this does not happen, there are always intermediates, and what we learned right now is just for theoretical value. Okay, theoretical value. But this kind of assumptions are not really valid in living system. All the pathways work simultaneously and do not take place one after another. Substrate. The substrates enter pathway and are withdrawn from it as and when necessary. So you can't be like oxaloacetic acid will wait until it gets converted to the next step. No. What will happen is whenever there is requirement, oxaloacetic acid will be removed from there and will be used somewhere else. It will be used somewhere else. So there is nothing like, citrate will become isocitrate. No. Citrate can be used in one more pathway suddenly. Do you realize? So there is always mismatch happening and there is no orderly fashion maintained in the body. Whenever there is requirement, intermediate will be removed and given somewhere else. Okay. And do not take place one after another. Okay. ATP is used as and when needed. It does not wait until everything is over. Enzymatic rates are controlled by multiple means, yet it is useful to do the exercise to appreciate the beauty and efficiency of the living system. See, the reason we are doing all this is to appreciate the beauty of it. Okay, it is not all waste. It is not all waste. You are doing it because you want marks, right? You are doing it because you want marks. I am teaching it because I love teaching, and such a beautiful thing this is, right? Yes. And in extraction or storing energy. Hence, it can be net gain of ATPs, 38 ATP molecules during aerobic respiration of one molecule of glucose, 38 ATP. But in the case of your anaerobic respiration, 2 ATP was there, 2 ATP, just 2 ATP was there, right? Now let us compare. Fermentation accounts for only partial breakdown of glucose, whereas aerobic respiration is complete degradation of CO2. In fermentation, the net gain of ATP is only 2 ATP. In fermentation, 2 ATP. But in the case of your aerobic respiration, 38 ATPs. Okay. Now, NADH is oxidized to NAD plus rather slowly in fermentation. However, it reacts very vigorously in the case of aerobic respiration. See, even NADH is partial. It knows, NADH knows I'll get more ATP in aerobic respiration, so it is acting and working very well. Whereas, same NADH in the case of fermentation, it is very slow. Like, nah, I don't want to do any work. It is like that. Okay. Now, students, no. Now tell me, in your house, if your mother has not made the food, what will you do? Tell me. Honestly, tell me. Don't tell me like, sir, I will cook anna sambar for an entire family. Don't tell me all that. Tell me what will you actually do if your mother is telling, I don't want to make food today. There are only two options available. One, either eat Maggie, either spend 2 minutes and make the Maggi. And the second thing, right? And the second thing is going to be ordering from Zomato or Swiggy. Yes or no? Tell me, yes or no? Tell me. You will either make Maggi or you will order from outside. You're like, it's fine. Dad is giving the money. I will order from outside, Zomato, Swiggy, order whatever you want and you'll eat. Right? You will order. So what is actually happening is, that was the backup, right? That was the backup. If the food is not made in your house, you went for a backup. Similarly, inside your body, if there is no carbohydrates, if there is no glucose, what will the body do? Tell me. Your body also has to go for a backup. Who is the backup in the body then? Can anyone tell me? Can anyone tell me who is the backup in the body? If there is no carbohydrates, if there is no glucose, if there is no fructose or there is no sucrose, what is the backup? Now students, in the first class, yesterday only, okay, first class I told you, there are other respiratory substrates. Such as your lipids, proteins, organic acids. Can they become the alternative now? Can they become the backup plan now? Tell me yes or no. Tell me. Can they become the backup plan? Yes, they can become the backup plan. So the backup plan, the call for backup is nothing but is breakdown of fats and breakdown of protein. Breakdown of fats and breakdown of protein. That is the backup. That is the backup in the body. Okay. So let's learn something called as amphiboic pathway now, and let's understand how your fats are broken down. Fats or lipids or glycerols are broken down. For example, right now I have fats. All of you know fats can be of two types. Fats can be of two types. For example, we have glycerol. Glycerol, and we have fatty acid. They will undergo beta oxidation. Now students, this is a PYQ, so listen very carefully. When fatty acids undergo oxidation, they will produce acetyl-CoA plus energy. Now students, tell me, can this acetyl-CoA right here, can it enter the Krebs cycle? Yes. Can this Krebs cycle, after this we have electron transport chain. After electron transport chain, as a result, can we have ATP? Yes, energy is produced. Energy is used. Right? Now, what about glycerol? When glycerol undergoes oxidation, it produces something called as DHAP. Dihydroxyacetone phosphate. This DHAP can isomerize into GAP and enter glycolysis. After glycolysis, what do we have? We get pyruvate. Pyruvate can become converted to acetyl-CoA. Acetyl-CoA can enter the Krebs cycle. After Krebs cycle, we have again electron transport chain. As a result of electron transport chain, can I get ATP molecules? Yes, I can. So students, do you realize from fats, which is a complex substance, we are getting ATP. We are getting ATP molecules. ATP molecules are getting. Okay? ATP molecules. Now, see here it is given here. Fatty acid, acetyl-CoA. We can also get FADH, NADH, which can be used in electron transport chain. Your glycerol can be converted to DHAP. Now students, what is the meaning of burning of fat? How many of you heard about this concept called as burn the fats? Today I will go to gym and I will burn the fats. What is the meaning of burning of fat, students? Tell me. And how can you scientifically achieve the burning of fat? Scientifically achieve. Tell me. How can you scientifically? It's a small nutritional fact here. The burning of fat means you're reducing the fat volume in your body, fat percentage in your body. Okay, fat percentage in your body. Students, understand, the only way the fats will burn in the body The only way fats will burn the body, tell me, only when there is call for backup. Unless there is a call for backup, can I tell the fats will be consumed? No. So if in the body there is carbohydrates, our body will use carbohydrates and survive. If I reduce the carbohydrates, students, if I reduce the carbs in my body, can the body be forced to burn the fats now? Yes, tell me. If the carbs are reduced in my body, if glucose is very less in my body, glucose or fructose or sucrose, sugar, that's why they say don't eat sugar much. Right? If you reduce the glucose in the body, what happens? If you reduce the glucose in the body will go for a backup. Body needs a backup. The backup is what? Backup is going to be fat. Backup. So you should never stop carbohydrates. First of all, you should never completely stop carbohydrates. Never stop it. Reduce the quantity of carbohydrates. Automatically, when you work out, when you go to gym or do something, as soon as the carbohydrate is consumed in your body, the fats will start to burn. Then. That is the science behind burning of fat. Okay. Clear? I did not know that. Okay, students, that is called as burning of fat. Now, what about in the case of proteins? In the case of proteins, obviously, if protein breakdowns, we get amino acids. Such as your glutamic acid, friends. This glutamic acid, if it undergoes deamination, it will produce alpha-ketoglutarate. Now, tell me, students, can this alpha-ketoglutarate enter the Krebs cycle? Yes. Enter the Krebs cycle. Once it enters the Krebs cycle, can it produce energy later on eventually? Yes. Krebs cycle, electron transportation, energy formation, it is happening. Now, one more amino acid, that is aspartic acid. Both acidic amino acids. Okay. Aspartic acid undergoes deamination. It can produce oxaloacetic acid, OAA. It can produce OAA. Now, this oxaloacetic acid, can it enter the Krebs cycle now? Enter the Krebs cycle? Yes. Enter the Krebs cycle, enter electron transport chain, make ATP, body is set. So students, the protein can also break down and give you energy. Not only carbohydrates and not only fat. Even a protein can break down and give energy, but this will happen very rarely when the protein levels go down completely. Okay? Clear, students? Aspartic acid. So that's why, students, you learn till now. Now, what do we learn right now? Right now, we learned that inside your body, proteins and fats are giving certain simple structures, right? Amino acid, glycerol, and everything. They're simple structures. These simple structures can also produce your energy, can undergo respiratory pathway and produce certain things. Even your normal substrate, it is glucose. Glucose can also break down and give you amino acids and fatty acids, right? So, can I call this from simple substance we are making complex substance? Simple substance to complex substance. Can I call it anabolic? Yes, it is anabolic. But here we have complex substance broken into simple substance. This is catabolic. Catabolic, anabolic. So, this is called as amphibolic. So, it is neither catabolic nor your it is not catabolic, or it is not going to be your anabolic. It is going to be amphibolic, because it is called as amphibolic. Why? It is called as amphibolic because both from simple substance we are making complex substance, from complex substance we are breaking into simple substance. Both the things are happening in the case of your bod plants and in your body. Okay, break you want? Wait, last topic I will tell you. Then we'll go to break. Before, okay. How much is left? 10 minutes and I'll give you a break. After 10 minutes, we'll take a break. Clear? After 10 minutes, I'll give you a break. Small break we'll take after 10 minutes. Let's read what is given here. Or we'll take a break now only, students. We'll take a break now. and we'll come back later. We'll finish amphiboic pathway, obviously. We'll finish amphiboic pathway, and then I will teach you respiratory quotient after break. We are only left with respiratory quotient and lots of questions. Lots and lots of questions. Okay, so let's finish this and then we will take a break. Set. Okay. Now, glucose is favored substrate for respiration. All of you know that glucose is the favorite child. Of the two brother and sister, the younger one is always favorite, right? Younger one is always favorite. Here also glucose is favorite always. Glucose is the favorite substrate for respiration. All carbohydrates are usually first converted into glucose before they can be used for respiration. Other substrates can be respired as been mentioned earlier, but they do not enter the respiratory pathway at the first step. No. Now, different substrate in the respiratory pathway. Fats would need to broken down into glycerol and fatty acid. If fatty acids were to respire, they would first be degraded to acetyl-CoA. See, fatty acid will get converted to acetyl-CoA, Fa, acetyl-CoA. Okay. Now, and enter the pathway. Glycerol would enter the pathway converted to PGAL or DHAP. PGAL or DHAP. Okay. Okay. PHA. The proteins would be degraded by proteases to individual amino acid, and after deamination, all of you learned, depending upon the structure, they would enter the pathway at some stage within the Krebs cycle. That is, they enter as oxaloacetic acid. Oxaloacetic acid they can enter, or they can enter as alpha ketoglutarate. Okay, or even as pyruvate or acetyl-CoA. This is called as amphiboic pathway. Since respiration involves breakdown of substances, the respiratory process has traditionally been considered as catabolic process. Traditionally, we have been calling it catabolic. Yes, metabolic. But in this understanding, correct, we have discussed above that at which point in the respiratory substrate, different substrate would enter if they were to be respired and used to derive energy. We just now learned that when fats break down, when proteins break down, we have oxaloacetic acid coming from somewhere. We have pyruvate coming in the middle. We have alpha ketoglutarate coming in the middle. They're entering the cycles from the, like wild in Big Boss, you know, in Big Boss there is a wildcard entry, right? That is the wildcard entry here, wildcard entry. Okay. What is important to recognize is that it is these very compounds that would be withdrawn from the respiratory substrate for the synthesis of the said substrate. Hence, fatty acids would be broken down to acetyl-CoA before entering the respiratory pathway when it is used as a substrate. Okay. But when the organism needs to synthesize fatty acids, acetyl-CoA would be withdrawn from the respiratory pathway for it. That is, meaning is whenever there is requirement, an intermediate is formed or intermediate is taken away. Are you understanding? See this line. When the organism needs synthesis of fatty acids, when it needs to synthesize fatty acid, it will use the CoA. It will use acetyl-CoA to make fatty acid. It will use acetyl-CoA when it needs fatty acid. But when the organism needs acetyl-CoA, it will break down the fatty acid itself. So based on the requirement, a particular molecule is broken down or is it made up? Okay. See, I'll read this line again. When organism needs fatty acid, what will happen? When the organism needs fatty acid, acetyl-CoA is taken up. But when acetyl-CoA is required, fatty acid is broken down. So whenever there is the requirement, things will change. Okay, respiratory pathway for it. Hence, the respiratory pathway comes into the picture both during breakdown as well as synthesis of fatty acid. To synthesize, we need acetyl-CoA. Formation of acetyl-CoA, we need to break down fatty acid. Two things are happening. Similarly, during breakdown and synthesis of the protein 2, respiratory intermediate forms the link. Breakdown process within the living organism is catabolic and synthesis is anabolic. Because respiratory pathway is involved in both anabolism and catabolism, it is called as amphiboic pathway. If you see here, students, this is the actual cycle happening here. Glycolysis, first pathway. Sometimes what happens is fat is broken down into fatty acid and glycerol. Fatty acid can converted to acetyl-CoA and enter here directly. Glycerol can be converted to DHAP and enter here. Amino acids can be converted to your oxalic acid or pyruvate and enter here. So this is the wild card entry. Directly they are entering from nowhere into the respiratory pathway. Clear? This is what is happening inside a cell. Now students, we will take a 15-20 minutes break. Now we will take a 20 minutes break, and after the break I want everyone to go eat and come. Run all of you and eat something and come. After the break, we will sit down and we will solve questions. What do you say? After you come back, all the way from the topics like glycolysis, the first part, from the first part to till now, whatever we have learned, from glycolysis to respiratory quotient, we will solve questions after the break. Okay. Now what is the time right now? This watch is my 5 minutes or 7 minutes fast, I don't know. 8:08. Okay, 8:08 is the time now. Class will resume at 8:28. Resume at 8:30. Let's take it at 8:30. Okay, 8:30 will resume the class. By the time I go out, 2 minutes will happen. Okay, so 8:30 will resume the class. Okay, 8:30 will resume the class. So I'll see you at 8:30, all of you. Don't run away. Stay back. Okay, go ahead and come. Yo, yo. Welcome back, students. Now we'll go towards the final end of today's chapter, and this has been really good fun and very fun chapter to teach. So, last concept right now is called as respiratory quotient. It's a very simple concept. Respiratory quotient is nothing but it is the ratio. It is the ratio between how much of CO2 is consumed, volume of CO2 evolved. In respiration, all of you know, CO2 is evolved as a byproduct. Yes, amount of CO2 evolved to the volume of CO2 consumed. That is called as respiratory quotient. That's all respiratory quotient. But this respiratory quotient can vary. Respiratory quotient can vary depending upon the different types of substrate. That is very important. For example, the respiratory quotient, it is CO2 evolved to CO2 consumed. Respiratory quotient depends. Let me erase this. Respiratory quotient depends upon the type of substrate. See, depending upon which type of substrate we are using in respiration, depending upon that, the respiratory quotient value, this value might increase or it might decrease depending upon the substrate. Okay. Okay. Now, when the carbohydrates are used, this is very important. For carbohydrates, when carbohydrates are used as a substrate, respiratory quotient is just 1, perfectly 1. Why? Because equal amount of CO2 is released to the equal amount of oxygen consumed. If 6 molecules of CO2 is released outside, it means that 6 molecule of O2 is consumed. And this is only 1 in the case of carbohydrates. Q. Now, next one is called as fats. Fats is actually less than 1. For fats, it is less than 1, that is 0.7. 0.7. Now, what is the meaning of that? That is, when fats are used for respiration, RQ is less than 1. Calculation of fatty acid is tripalmitin. See, students, in examination, they will never give you as fats. They'll give directly as tripalmitin. You should know tripalmitin is the fatty acid. It is a lipid. So, RQ will be what? Around 0.7. Can you see? More oxygen is being consumed. Less CO2 is out. Now, protein is 0.9. Proteins is 0.9. Proteins is small table, write down. Question can come from here. Okay. See here. When the proteins are respiratory substrate, the ratio would be about 0.9. What is important to recognize is that in living organisms, respiratory substances are often more than 1. Pure proteins or fats are never used as respiratory substrate, because when we eat the food, we are taking carbohydrates, we are taking lipids, we are taking proteins, everything is going inside. Not just pure protein is never going inside. There's always mix and match of things. Okay. So with that being said, the chapter is over. The first question is on your board. I know the question might not be visible to some of you because it is very tiny. Let me read it for you. The question is very simple. Understand? In plants, sucrose is converted to. Sucrose is converted or broken down to 2 molecules. What are the 2 molecules? Sucrose is a disaccharide. It is broken into 2 monosaccharides. What are the 2 monosaccharides? That is the question. Is it glucose and glucose, glucose and fructose, glucose and galactose, fructose and galactose? Go. What is the answer? I'll just move aside and let me refresh here. What is the answer? Students, every one of you quickly smash that like button and share this video as much as possible. Obviously, students, we know B for Baswa sir that glucose and fructose is present in the case of your sucrose. Glucose and galactose, students, Madhumita, that is for lactose. That's for lactose. Okay. You have learned this in biomolecules. Carefully, you have learned it. Okay, base of sir now. Now, next question. In glycolysis, the conversion of 1,3 bisphosphate to 3-phosphoglyceric acid. This is actually the seventh step of glycolysis. Is it energy yielding, energy utilizing, phosphorylation, isomerization? What is happening? Tell me. The seventh step. Seventh step. Energy yielding, you get energy here, ATP is produced in seventh step. Seventh step and the 10th step, ATP is produced. So it is energy yielding process, energy yielding phosphate, energy yielding process. Okay. No. Next question. ATP is used in 2 steps. All of you will tell me, sir, ATP is used in 2 steps. Which step? First step and third step. ATP. Students, let me tell you, the glycolysis which I have taught you, please practice that glycolysis. Unless you practice, and yesterday I gave you all the possible questions from glycolysis. Practice all those questions also. Once you do the practice of glycolysis which I have taught you, trust me, all of these questions will become very easy. Okay. Now, first ATP is used in converting glucose to glucose phosphate. Remember, glucose is there, one phosphate is added to this. Glucose is becoming glucose 6-phosphate. Okay. And the second is used to convert fructose to fructose 6-phosphate. Yes. Both are correct. It is explaining, yes. It is used in first 2 steps. And it is explaining where the 2 steps are. So what is the answer? Option number 2. Correct explanation of A. It is correct explanation of A, right? Realize, it is the correct explanation of A. Here it is telling ATP is used at 2 steps. Here it is telling you where the ATP is used. Like how the ATP is used. Right? How the ATP is being used. Option number 2, not 3. Option number 2. Okay. Next question. The conversion of glucose to glucose 6-phosphate. The first step. The first irreversible step. That is, we have 1, 3, 10. Right? Irreversible step. Glycolysis is catalyzed by. What is the name of the enzyme? What is the name of the enzyme involved? What is the name of the enzyme? I've told you. Always remember the first enzyme and the last enzyme. First enzyme and the last enzyme, always. Okay. What is the answer? Hexokinase. Beautiful. Hexokinase is the answer. What is the role of NAD plus in cellular respiration? What is the role of NAD plus in cellular respiration? It was a NEET 2018 question and only 50% of students, almost 60% of students were able to answer this question. Difficult question, understand. Does it function as an enzyme? No. It is a nucleotide that is source for ATP synthesis? No. Okay. Final electron acceptor? That is no. Is it? It is the function as electron carrier everywhere? Yes. It is functioning as electron carrier everywhere. It is receiving the electrons, undergoing oxidation everywhere. Electron carrier, students, electron carrier. Your famous NAD plus. Okay. Next question. Sucrose is converted to glucose and fructose by an enzyme. Which is that enzyme, students? Which is that enzyme? How do you convert sucrose into glucose? The enzyme is called as invertase enzyme. I told you. In the case of plants, invertase enzyme. Yes, invertase enzyme. That is sucrose is converted to glucose in the presence of invertase. Invertase enzyme. Yes. Hi. I can see hi. Hi. Pushpendra Kumar. Hi. Now, next question. Glycolysis is found in cytoplasm of virtually all type of aerobic and anaerobic cells. In this process, glucose is converted to. This is a very easy question. Quickly tell the answer. Everyone, quickly tell the answer. Don't take so much of time on this. Done? 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up. All of you know pyruvic acid or pyruvate. Pyruvic acid or pyruvate. All of you know. This is a very easy question. There's thinking is not required for this question. Okay. Next question. In which of the following reaction, glycolysis, a water molecule is removed? All of you remember, students, in the ninth step, you remember, water is being removed. Tell the answer now. Tell the answer. In which step water was being removed? The ninth step. That is the clue. Ninth step, water was being removed. Ninth. This is the last step. This is the last step. Don't. This is not the thing. Glucose glucose. There was ATP was produced. No water. Fructose to fructose 1 bisphosphate. Again, phosphorylation. No water removing. But students, in the 2-glyceraldehyde phosphate to phosphoenol pyruvate, did we remove water? Yes, we removed water there. Yes, we removed water here. Now, choose the wrong statement. CO2 is harmful molecule and releases during catabolic reaction. Okay, correct. Respiration will always require O2. Breathing is a part of respiration. Yes, correct. All of the above. Which is the wrong statement, students? Which is the wrong statement? Time starts now, go. Time starts now, go. Which is the wrong statement here? Wrong statement. CO2 is harmful, yes. The students tell me, does the respiration always require O2? For example, the first step in respiration, the glycolysis. Haven't I told you, glycolysis can take place both in aerobic as well as anaerobic reactions, anaerobic organisms, both. Yes, both. Do you remember? I have told you respiration, that is first step, glycolysis can happen in both aerobic and anaerobic, both. So can I tell the second statement is wrong? Yes. Second statement is wrong. Wrong statement. Wrong statement, students. All of you should know CO2 is harmful and is removed during catabolic reaction. It is removed. Breathing is part of respiration. Yes, breathing is part of respiration because when you breathe, oxygen enters. Oxygen is entering inside and that oxygen is used for respiration. Right. So second statement. Next question. Which of the following is not correct regarding glycolysis? Not correct. In the word as written from Greek was glyos, yes. Okay. The scheme was of glycolysis was given by Gustav EMP pathway. Yes, it's called as EMP pathway. In all organisms, it is the only process of respiration, students. Tell me, is this the only process of respiration or are there any other thing also? Do we have Krebs cycle? Do we have fermentation, alcoholic, lactic acid, electron transport chain? Tell me, there are other things also, right? There are other things also in case of respiration. So glycolysis is not the only process of respiration. There are other following also, process also. So 4 is the answer. Students, do you realize the concept is very easy? The questions can be tricky. Are you feeling that? The questions are tricky little, but the concept is very easy. So that's why I tell you, students, that's why I tell you, try attempting more and more questions. Join a test series as soon as possible. Join a test series and start solving questions because unless you solve questions, the backlog will never clear. You cannot say Tata bye-bye to the backlog unless you solve questions. So solve as much as question as possible, then only you can confidently tell, yes, the backlog is over. So every single day try to solve at least 50 to 60 questions. Keep on solving questions. Don't take break from that. In fact, look at this next question. Find the odd one out statement. See, odd one out. Glucose is derived from the end result of photosynthesis, that is sucrose. Glucose is coming from sucrose, yes. Invertase acts on sucrose to release glucose and fructose, yes. Glucose and sucrose are phosphorated in the presence of hexokinase. The steps of glycolysis from the third step onwards are same for glycolysis and fructose. Yes, it is also correct. It is also correct. Tell me which is odd one out now. Which one is odd one out? This is a very tricky question. See, it's a beautiful tricky question. See, after the third step, when isomerization happens, when isomerization happens, yes, it is same. Glucose and fructose, it is same to same. Why 3 is the answer? Why third one is the answer? The third one is the answer because it says sucrose also, students, sucrose. Glucose and sucrose are phosphorated in presence of hexokinase. No, only glucose is phosphorated. I repeat again, only glucose is phosphorated in the presence of hexokinase. Your sucrose will be broken down. Then that glucose will enter the cycle. Sucrose will never enter. So do you realize it was such an easy question? It may seem easy, it may seem difficult, but it's a very easy question. Very easy question. Next question. Students, take your time and answer. Take your time and answer. It's a very easy question. All of you know this. You can answer this very easily. So take your time. All of you know glycolysis, cytoplasm. Citric acid cycle, mitochondrial matrix. Electron transport system happens in the inner membrane. Proton gradient in the intermembrane space. Intermembrane proton gradient. So what is the answer now? Tell me. Tell me what is the answer then? What is the answer? A is 2, B is 1, C is 4 and D is 3. Option number 1. Option number 1. A. Exactly. Option number 1. Next question. Students, I feel all of you are already saturated. I don't know why. I feel all of you are very tired by the end of the day and there is no motivation to study. There is no motivation to solve questions. Bring out the energy, students. You have hardly left around 30 days you have. 230 days you have, students. Unless you bring the energy right now, you will, all this you will not be able to do towards the end. Do right now only, do continuously. Okay, show the energy. Now, identify the step in tricarboxylic acid cycle which does not involve oxidation. I did this question already. We did this question already. The second option, because here GTP is formed. GTP is formed. GTP is formed here. Yes. Next question. Read the question and see. Read the question. Read the question, all of you. Read the question. Take your time. Take your time, all of you. Take your time. Take your time, all of you. Proud to be plants, huh? All of you are plants now. Be like plants. Now, which of the following statements are correct about respiration? Correct about respiration. Okay. Energy of oxidation reduction is utilized in phosphorylation. Yes. Everywhere oxidation reduction energy is used. Correct. Oxygen acts as final hydrogen acceptor or electron acceptor. Yes. Photooxidative energy is used for production of proton gradient required for phosphorylation. No. We know photooxidative energy is used in photosynthesis. It is used in photosynthesis, not here. Photooxidative energy is used in photosynthesis and not in respiration. This statement is wrong. Statement is wrong. Okay. Now, the role of oxygen is limited to the terminal stage of the respiration. Yes. Only terminal stage it is using. Okay. No. A is correct. A is correct because oxidation reduction energy is used. Okay. Proton can cross outer membrane of mitochondria through a channel formed in the integral membrane proton complex. No, they cannot enter like that. So what is the answer? A correct, B correct, D correct. Even if you don't know this last option, you can still answer this question because wherever we have A, B, D, there's only one option here, A, B, D. Because C is wrong, we know, C is wrong, we know, C is wrong. So 2 is the only answer. With the help of elimination method, you can. Photosynthesis, yes, I will upload. I'll upload, don't worry. Soon I'll upload photosynthesis. In fact, today only I'll upload, don't worry. Or tomorrow morning I'll upload notes. Next one. Take your time and answer again. Take your time, answer this question. It's a PYQ from NEET 2023. NEET 2023 question. Take your time, all of you. Take your time. Oxidative decarboxylation, glycolysis, oxidative phosphorylation, tricarboxylic acid. Tricarboxylic acid cycle. Take your time. I'll give you 1 minute. Aram se think and then answer. Take you 1 minute time you take. Such questions in examination, yes, in pressure, you might go wrong, but right now you have time. Okay, right now you have time. Think and answer. I want everyone to try. It doesn't matter if you're going wrong, doesn't matter if you're right. Keep trying. Unless you try, you don't know that, if you're right or wrong, right? So try. It's a simple match the following. Simple match the following answer. Simple match the following answer. Okay. Now students, let's see here. First we have oxidative decarboxylation. It happens in link reaction, and the enzyme involved in link reaction is pyruvate dehydrogenase. Okay. Glycolysis. Glycolysis is where is glycolysis? Glycolysis. It's called as EMP pathway. Yes, it is called as EMP pathway. And then Mayer of Parnas pathway. Oxidative phosphorylation. Phosphorylation is electron transport chain. Electron transport system or electron transport chain. Carboxylic acid is citrate synthase. I told you. Learn the first enzyme. Learn the first enzyme. So what is the answer now? AS2. AS2 means this is gone. This is gone. B is 4, right? B is 4. This is the answer. Don't even need to look at other options. Don't even look at other options. B is 4. Next question. Okay. How many times decarboxylation occurs in each TCA cycle? Very easy question. Everyone should answer. Time starts now. Go. Very easy. Very easy. How many times decarboxylation happens? Decarboxylation. 1 time, 2 time, 3 time, 4 time, 5 time, 6 time, or it keeps on happening? Tell me. Decarboxylation. How many times it happens? Entire decarboxylation. Tell me. It happens twice. All of you know. 2 times it happens. 2 times it happens. Only 2 times. Right? Not 3 times. 3 times will happen if you include the link reaction. If you include the link reaction, then it will happen 3 times. Answer is twice, option number 4. Twice, option number 4. Okay. Next question. Fatty acids are connected to respiratory pathway through. How are fatty acids connected to respiratory pathway? Amphiboic pathway, students, understand. Amphiboic pathway. How are fatty acids entering? Fatty acids are undergoing something called as beta oxidation to produce something called as what? We learned this right now. 2 minutes, 5 minutes back only we learned it. Fatty acids are connected to respiratory pathway through fatty acid, students. Fatty acid, not glycerol. Not fatty acid, not glycerol. See here. Forgot already. Oh, we read already so many questions. Fatty acid is connected to acetyl-CoA. Glycerol, DHAP. Glycerol, DHAP. What is the answer? What is the answer? Now tell me. Obviously, it's the. What is the answer? Now tell me. Acetyl fatty acid is converted to acetyl-CoA. It's converted to acetyl-CoA, students. DHAP is for glycerol. Glycerol. This is for proteins. Alpha ketoglutarate acid is again your proteins. This is for fatty acid. Fatty acid. Okay. Next question. What amount of energy is released from the glucose during lactic acid fermentation? I told you all yesterday's class. It is very less, 2 ATP. Just 2 ATP. Which was less than 7. Less than 7. NCERT line. NCERT line. NCERT line less than 7. Students, tell me, what is the maximum amount of maximum concentration of ethanol produced by yeast? Maximum concentration, tell me. Maximum concentration was 13%. 13%. It cannot go above 13%. It cannot go above 13%. I told you yesterday's class. 13% was 13%, not 15. 13. Okay. Next question. Take your time and answer again. Take your time and answer. Time starts now. Read the question. Complex are given here. Names are given. Some of them are they have given the inside filling inside the complex also. What is the electron carriers is given. Read the question. Take your time. 1 minute and your time starts now. Go. Take a minute at least. Take a minute at least. Take a minute and time starts now. Go. What is the answer, friends? What is the answer? Complex 1 is called as NADH dehydrogenase. Complex 1 is called as NADH dehydrogenase. So can I tell these two are gone? Yes, these two are gone. These two are gone. These two are gone. Okay. NADH dehydrogenase. So A is going to be 3. Complex number 2. Complex number 2. Okay, complex number 2. Can I tell complex number 2 was called as what? FADH dehydrogenase or succinate dehydrogenase. FADH was used in complex number 2. Complex number 3. Complex number 3 was called what? Cytochrome BC1 complex. BC1. What is left now? Complex 4 is going to be, complex 4 is cytochrome oxidase, which has your copper centers. 2 copper centers, that is copper center A and copper center B, and we have A1 and A3. A1 and A3. So 4. Exactly. Next question. Read the question, very easy. Quickly tell me the answer. Do not take more than 10 seconds. Do not take more than 10 seconds now, all of you. Sanatra, Savita, Riz, Madhumiti, Sumi, Tanuja, Pradeep, Sukanya, Serotonin, all of you. Very easy question. Shoot your answers. Identify the cytochrome which act as a mobile carrier for transfer of electrons from complex 3 to complex 4. From here to here it was taking. From here to it was taking. From here to here. That is cytochrome C. Very good. Cytochrome C. From complex 3 through complex 4. Complex 3 to complex 4 it was taking. Nicely traveling and going. Okay. Next question. The number of times decarboxylation of isocitrate occurs during TCA cycle. Okay. Students, sometimes what happens is they will make a tricky question like this. They will make a tricky question like this again. See, now time isocitrate is undergoing oxide decarboxylation. All of us know what is the trick here? Officer, can I keep selling? See, here we have citrate, here we have isocitrate. Okay, sandwiches. Isocitrate is here. Now tell me, students, where does the first decarboxylation happens? First decarboxylation happens between isocitrate and alpha ketoglutarate. First one is happening here. Second one is happening between alpha ketoglutarate and succinyl coenzyme A. Second one is happening here. What is the answer then? What is the answer? Since it is happening after isocitrate, 2 are also happening after that. Yes. So 2 answer. Students, do you realize sometimes what happens? You know the answer, they can make it a little tricky to trap you. Hopefully you don't get trapped. Okay. The 5-carbon compound formed in TCA cycle is, which is the 5-carbon compound? See, everything is 6, but after the first decarboxylation, we get 5-carbon compound. the first decarboxylation. That is nothing but alpha ketoglutarate. That is nothing but your alpha ketoglutarate. After the first decarboxylation, alpha ketoglutarate. Next question. Which of the following statement is incorrect? Incorrect statement. Incorrect statement. Take your time, read the statements, answer confidently. We are cunning fox, huh? Yeah, sure. Sure thing. Read the question now and answer. Take your time. Find the incorrect statement. Incorrect. Which is not correct. Done? Done, students? See here. ATP synthesized through complex 5. ATP synthase, yes. Oxidation reduction reactions produce the proton gradient in respiration, yes. During aerobic reaction, role of oxygen is limited to the terminal stages. Until the very end, your oxygen is not entering anywhere. In electron transport chain, one molecule of NADH is giving 2 ATP? No. One molecule of NADH is giving 3 ATPs. And one FADH is giving how many ATP? 2 ATP. Not 3 ATP. Not 3 ATP. So what is the answer? Now tell me. What is the answer? ATP is synthesized through complex 5, right? All of you remember complex 5. The F0, F1 complex, complex 5, complex 5. Proton is sent up, electrons are sent up, protons are being sent up. 4 H plus are going up and 1 ATP is coming out. 1 ATP is coming out. Okay? Easy question. Okay. Very easy question again. The number of substrate level phosphorylation is 1 turn of citric acid is. How many substrate level phosphorylation? That is, how many GTP is made? GTP is nothing but substrate level phosphorylation. Where is GTP made? Where is GTP? How many GTP is made? Only one GTP, right? Between succinyl coenzyme A to succinate. Succinyl coenzyme A to succinate, one GTP is made. One GTP is made. Respiration coefficient for palmitin, tripalmitin. Tripalmitin is a lipid, so it is 0.7. 0.7. All of you know this, 0.7. This is your homework question for the comment section. Homework question. Don't forget to write the homework question, students. It is very difficult for studying 3 hours and teaching and then making sure that all students are understanding every single concept, clearing the doubts and finishing the chapter. So with that, students, the chapter is actually over with practice of questions and you have realized where you are going wrong also, right? You have realized where you are going wrong. Don't tell him now. Don't tell now. It's fine. I will not see. I will close my eyes. Don't tell the answer now. In the comment section, answers. Okay, I will not see the answers in the chat. Okay. So students, that was the end of the session, today's session. And I feel this week, right? This week was very productive. This week it started off with biomolecules part 2 we did, right? Biomolecules part 2. Where we learnt so many new things, enzyme reactions and everything. And later on we learnt about respiration plants, both the part 1 and part 2. And if you notice, we have done more than 9 hours of teaching on this channel itself, right? And weightage of this particular chapter is around 4 to 5 questions. 4 to 5 questions will come, or 6 questions sometimes. 6 questions can come. Okay, so that is the weightage of the chapter because mineral nutrition and transport implants has been removed, right? Since those chapters have been removed, weightage of these chapters have been increased now. So this is the core plant physiology, right? So students, thank you so much, students, for today's class. Let me know in the comment section. I will read the comments and I will heart it also, and I will give a like to it also. How was plant physiology with me, right? Did you understand Krebs cycle? Did you understand electron transport chain? Did you understand what is oxidative decarboxylation? Did you understand what is glycolysis? Not by hearting any of it. Not by hearting any of it, right? So yeah, understand and let me know in the comment section how was today's class. And students, those of you who have not still registered for MESAT, link is in the description. Quickly join the MESAT because you can get into Vedantu's paid platforms very easily, like 70% off, 90% off. If not that, you can also get iPad, iPhone, there are drones, PS5, so many things are there. So go grab it. Okay. Sir, sorry for so much late. Soumya, it's fine. You watch recorded only. You watch recorded only, it's fine. Now class is only over. Okay. So thank you so much, friends, for today's class and have a great day ahead. Sleep well, drink a lot of water, and revise. And revise. With me, NCERT reading is done. You need to revise your NCERT on your own also one more time. Okay. So thank you so much, students.